Okay. Hello, everyone. Thank you for being here. Thank you for waiting. It's 8 minutes since the beginning of the webinar. We have waited for people to join us, also in presence, because today we are here at the Politecnico di Torino. I am Andrea Spotti. I'm part of the Reborn consortium with the T-Next Innovation Hub. We take care of the dissemination and communication of the Reborn project. Along with me today, there are Dr. Giorgio Montalbano and Professor Chiara Vitale Brovarone, who's project coordinator of the Reborn project. And today they are going to present the first webinar of the Reborn project, which is part of a series of webinars they were going to be holding during the next months to present the project consortium work and the results achieved so far. The first part for the introduction will be held by Professor Vitale Brovarone, and then Giorgia Montalbano will then come a little bit more into the details of the project. I would leave the floor to you, Chiara, and thank you all.
Thank you. Good afternoon, everybody. Thank you, Andrea, for the nice introduction. I will give you an overview of the project. Okay. So my name is Chiara Vitale Brovarone. I am full professor of material science and technology at Politecnico di Torino, and I am the coordinator of the Reborn project. So here in this slide, you have some data related to the call under which the project has been funded. The type of action is a RIA, so a research and innovation action, and the duration is... Okay.
Sorry, just 1 second. We just need to share the screen. Apologize for the interruption.
Okay, thank you, Giorgia. Okay, so you have here some data related to the call under which the project has been funded. As you can see, is a research and innovation action area. It lasts 4 years, and we are now in the last year of the action. Here you have a map of the consortium partners. As you can see, there is a perfect balance between the research partner and the industrial one, and we have organized the workflow in order to continue to support each other and in order to synergistically combine our expertise and our perspective in order to maximize the project impact. The Reborn project is framed in the context of myocardial infarction. As you know, a myocardial infarction occurs when the blood flow is hindered or blocked in an area, and this blockage leads to ischemia and necrosis of the heart tissue. We are mammalian, and as all other mammalian, our heart cells have a very limited regenerative capacity. So after myocardial infarction, there is a loss of cardiomyocyte that became ischemic, and this is quite severe in its impact. After myocardial infarction, as after every type of injury, the reparative action of our body begins, and this begins with an inflammatory reaction. Unfortunately, this inflammatory reaction is quite often too extensive, both in the area and in the timing, and so it is persistent. When we have persistent inflammation, we can observe the proliferation of myofibroblasts, and these in turn can lead to the formation of a rigid scar tissue, and the formation of this rigid tissue can lead to ventricular dilation. So as you can see, after myocardial infarction, you can have a full chain of events, and these events are called adverse remodeling. This event can lead to heart failure. Heart diseases in Europe account for a large number of millions of deaths almost 4 million, and heart diseases are the #1 killer of both women and men in the majority of the European countries. In this context, the project ambition is to provide a solution to, let's say, reduce the occurrence of adverse remodeling. So what we are planning is to develop and provide to the population a multifunctional smart cardiac patch that will be developed at the lab and industrial scale during the Reborn project, and that hopefully after the end of the project could be translated to the clinic in order to prevent heart failure in patients that survive a myocardial infarction, thus reducing their mortality and enhancing their quality of life. As you can see, they should be happier with our patch. So overall, our concept combines a set of multifunctional carriers. These carriers have the goal of controlling inflammation, inhibiting the fibrosis, and promoting cardiomyocyte proliferation. But these functions are not the only one that we want to provide. We want also to provide, through the technique that we are going to use, the electrospinning, a topological stimuli to the heart in order to promote a more physiological-like regeneration. So this is achieved by the use of electrospinning and also by the use of a polymer that is a piezoelectric one and that can enable the electromechanical coupling with the heart tissue. All this concept is meant to be delivered through minimal invasive surgery in order to address the different aspects that need to be modulated in order to prevent the adverse remodeling of the heart tissue. So here are our objectives. We want to develop a multifunctional piezoelectric patch that will be acellular, so it will not include cells, and it will provide stimuli that are meant to be anti-inflammatory, anti-fibrotic, and to exert a cardiomyocyte proliferative effect. The patch, as I was saying, is meant to be delivered during minimal invasive intervention, for instance, during coronary artery bypass grafting surgery, CAB. The patch and all this multifunctionality will be, let's say, enhanced by the use of an ultrasound stimulation platform. The goal of the US stimulation platform is to exert a control on the drug release kinetics and mechanism to promote the penetration of the drug within the myocardial tissue, but also to exert per se an effect on the heart cells. All along the development of the multifunctional patch, we have a few partners that are working in the development of an advanced in vitro model that will support the validation of the patch, but will also be a key exploitable result per se, because it is much more advanced than the current in vitro model available in the scientific panorama. So here I have summarized the 5 specific objectives of the Reborn project. The first one, of course, is related to the development of the US platform. As you will see, there will be 2 different US platforms developed within the project. The second one is related to the development of several types of carriers and their incorporation within the electrospan piezoelectric patch. The third one is completely devoted to the development of this advanced in vitro heart chamber model. The fourth one is related to the transfer from TRL3, which means lab scales, toward TRL5, which is the industrial scale, both for the patch, but also for the heart chamber technology. And the fifth one is linked to the preclinical validation of the multifunctional patch in both small and large animal models. So in order to accomplish all these set of specific objectives, we have organized the work in these 4 years in the frame of 4 different work packages. Of course, there is a work package that is related to management, but also to the risk assessment and mitigation strategies, as well as ethical issues. And this is work package 1. Then we have the last work package that run in parallel along the project duration, which is related to the business case development, so the potential industrial exploitation, but also the dissemination and communication activity as the one that we are having and holding today. And then you have the core of the project, other 6 work packages that are divided, let's say, into different stages or stage of development. The first 3 are related to TRL3, so more at the lab scale, whereas the last 3 are related to the translation activity, so a TRL5. And I will go very briefly into each of these 6 work packages. Of course, in order to make things working, there is a lot of effort from the people staying in the lab, supervised by Georgia, the task leader, and the work package leader. And here you have the representative of all work packages, coming both from the academic and from the industrial panorama. So coming back towards the work packages 2 is related to specification, requirement, and benchmarking. And basically in this work package, we have analyzed all the chain of events occurring after myocardial infarction. So what is happening and in which time scale, in order to identify the drugs that should be incorporated within the carrier and the patch in order to control inflammation, reduce fibrosis, and promote cardiomyocyte proliferation. So let's say a brainstorming, analysis of data phase, and some requirement that has been fixed in the first 6 months of the project. Then some partners have developed health economic modeling and identified the cohort of patients that would benefit the most from the Reborn patch in order to better address our innovation. And then we have defined, as far as the heart chamber is concerned, the specification of what this heart chamber model should be able to do, basically. Work package 3 was really the core of these first 3 years. In this work package, several partners have worked extensively in order to incorporate the drug that we selected, ibuprofen as anti-inflammatory drug and pirfenidone as antifibrotic drug. This drug has been incorporated whether within the carriers or within the fibers, so the carrier that has been then dispersed in the fiber, or the drug directly within the fibers. And another partner has worked a lot on engineering extracellular vesicle containing microRNA cluster able to promote cardiomyocyte proliferation. The technology that we have chosen to develop the patch is electrospinning that has been implemented both in its uniaxial and in its coaxial configuration, but on which Georgia will go much more into detail. In work package 4, we have performed all sorts of in vitro tests. This has been more, let's say, conventional, using macrophage and cardiac fibroblast, both on the carrier and the patch, with and without ultrasound stimulation. We have also evaluated the engineered extracellular vesicle in their ability to promote cardiomyocyte proliferation. And the 2 partners working more closely on the heart chamber concept has been developing these structures that are basically electrospan structure that are then mounted on this bar and are then filled with a gel and cells, and then they are positioned into a perfusion dynamic culture environment in order to mimic a pumping heart chamber. Coming to work package 5, here we have the translation and demonstration. The first images are related to the heart chamber concept, positioned within its perfusion dynamic environment. The second one is related to the industrial translation of the electrospan patch at BioInitia, which is one of the partners of the project. And the third one is related to the translation of all the material and patch and concept into a medical device, not yet on the market, of course, but translated at the industrial scale by our partner Corsim. Last but not least, work package 6 related to the preclinical validation on the patch produced in collaboration between Politecnico di Torino and BioInitia. This will be tested in the next month in mice, and then the best performing prototype in landrace pigs, and this will be performed by stimulating with the ultrasound platform. As I was saying before, the partner in charge of the ultrasound platform, Scuola Superiore Sant'Anna, first developed a platform for the lab scale test, and then a second platform supported by a long modeling phase to be used in vivo in order to be sure that the right dose and intensity of ultrasound will reach the myocardium, taking in consideration all the reflection by the human body tissue. So this was really a quite significant modeling work. Last experimental work package is related to what? To the clinical specification. So what will be the next step after the ending of the project? If we really want to go into the clinic, what will be the test that we have to perform in phase 1? So proving the safety and feasibility of our approach, and into phase 2, so proving the efficacy and the long-term effect of our patch. Well, we have a partner in Porto in the cardiovascular unit that is trying to figure out what could be the following steps. Then we have another partner located in Berlin that is using the Markov model. And in this case, they want to compare the benefit and quality of life of patients undergoing standard of care procedure or undergoing standard of care procedure plus the implantation of the Reborn patch, in order to have a clear idea of the value of our innovation. Last but not least, the partner that is already in the cardiological market with the other products is dealing with the regulatory strategy and the dossier template file. So with this, I have closed my short presentation and introduction to the project. We can have a look at the project video, which is again general, and then we can go with Professor Montalbano into the detail of what has been done. Thank you very much for your attention. Okay, so thank you very much for your attention. We can now move to the more technical presentation by Professor Montalbano.
Okay. Good afternoon, everyone. Just a few seconds that I share my screen. Okay. Fantastic. So good afternoon again. So welcome to everyone. My name is Giorgia Montalbano. I'm working as assistant professor here at the Department of Applied Science and Technology of Politecnico di Torino, here together with in a group of professors led by Professor Chiara Vitale Brovarone, and I'm an active part of this really challenging project. And so after having an introduction about the general objectives and vision of this project, let's focus more on the different multifunctional biomaterials and reborn technologies that were developed in order to design the cardiac multifunctional patch. Chiara has already presented the large number of technologies that have been developed so far in order to meet very key and important aspects in the design of a smart multifunctional cardiac patch meant to the support of the functional regeneration of cardiac tissue after the myocardial infarction. And so today we will go through all the strategies that were successful and that were optimized and integrated in order to enable the design of the patch. So in order to meet in the design very key features such as the electromechanical coupling with the cardiac tissue, a nanostructure that can be biomimetic and also highly flexible, the high biocompatibility of all the components and the overall and the entire patch, and of course the opportunity to have a simultaneous delivery of different therapeutic agents that can in some way target all the different phases of the regeneration of the functional cardiac tissue. So if we focus on the first main characteristic that we require for the design of our patch, so the fact to have a very a good electroactive substrate, we immediately looked at a very interesting class of smart material that is represented by piezoelectric polymers. So why we choose this class of material? The reason is based on their intrinsic ability to generate an electrical charge when subjected to mechanical stresses and deformation that are due to the molecular dipole moments reorientation. So it means that this kind of material can potentially promote the electromechanical coupling with the aim to restore the compromised transmission of the electrical signal through cardiac tissue after MI. So between the large number of piezoelectric polymers that we have available for biomedical application, for sure, polyvinylidene fluoride represent one of the most well-known and studied, especially also due to the high biocompatibility, the high chemical resistance. In fact, is already found large use in the clinical practice. Polyvinylidene fluoride is characterized by a semi-crystalline complex structure presenting 5 distinct crystalline phases, where beta phase is the one responsible for the piezoelectricity of the material due to this polar transplanar zigzag conformation of oriented hydrogen and fluoride unit cells, along the carbon backbone. So this means that acting on the final content of the crystalline phase of this polymer, we can modulate the piezoelectric properties of the final substrate. And this can be done mainly following three main routes represented by mechanical stretching, electrical pulling, and the introduction of bulky comonomers. But where the last option may in some way lead to compromise, may compromise the biocompatibility of the overall polymeric system, we can exploit the synergistic effect of the mechanical stretching and electrical pulling by processing this polymer by using a well-known advanced manufacturing technique, that is the electrospinning technique. So the electrospinning technique is a manufacturing process that is based on the extrusion of a polymeric solution, a suspension or fuse that can be subjected under the application of a high voltage electrical field that is stretching the polymer into the formation of a polymeric jet that is then solidify in a nanofiber when travels toward the grounded collector. So this technique is a very versatile and scalable manufacturing process that is used mainly to design macro and nano nanofibrous structure that can in some way mimic the features, the morphological feature of the extracellular matrix, and also is characterized by a very high surface area and high porosity that is a real advantage in many applications. So moreover, we can also tune the final morphological, chemical, and mechanical properties of this substrate just according to the defined experimental setup and the properties of the material formulation. So here in the slide, you can see the series of options that we can, the different combination that we have in our experimental setup and the different type of material that we can obtain exploiting this versatile technique. So just changing some components of our setup. So if we focus on the upper part of our slide, we can see how just changing the collector, we can in some way modulate the different orientation of the deposited fibers, where using plate collector, we obtain randomly oriented fibrous matrices, while when we use drum rotating collector, we favor the alignment of the deposited nanofibers. And also, if we even use very small diameter drum collector, called mandrels, we can also reach the design of tubular structure. Looking at the material formulation, if we can also introduce some inorganic phases into the polymeric formulation that can be processed via an uniaxial system in order to promote their homogeneous distribution or the alignment into the fibers, or we can even exploit the simultaneous processing of two different polymeric systems using coaxial spinners in order to combine different materials and finally obtain core shell fibers. So in Reborn, we have optimized and explored these very interesting and versatile processing technique, mainly to address some key aspects in the improvement, not only improvement of the manufacturing process in terms of stability and versatility of the overall electrospinning process, but also toward the use of lower toxic solvent compared to the gold standard for the electrospinning. But also in a view to improve the piezoelectricity of our materials, so in order to control the crystallinity content and increase the electroactive phase, and also to induce the alignment of the deposited fibers in order to obtain more biomimetic construct. So starting from the material formulation, as you know, even if despite the great advantages represented by this technique, one of the main limitations is represented by the large use of very toxic solvents that are very effective in the processing of the polymeric formulation, but can be harmful, of course, just not only for us and for the environment. So one of the goals in our project was to replace the use of these toxic solvent with some less toxic mixture. And so in this case, we decided to select a mixture of acetone and DMSO in order to replace the most commonly used dimethylformamide, normally used for the processing of PVDF. So based on this choice, we then optimize all the other process parameters. So mainly the polymer concentration, of course, were optimized according to the fact that we need a very stable process for a long time. So we combine these optimized parameters with all the other process parameters in order to properly balance the applied voltage, flow rate, needle diameter, and needle collector distance, where the collector, we selected a drum rotating collector in order to induce the alignment of the deposited fibers. So thanks to the optimization of the run process and the combination and the study of the combination of the different process parameters, we could end with a very stable process over time that led us to the design of a self-sustaining PVDF membrane. As you can see here from the images, these membranes were characterized by a highly aligned fibers with an average diameter of about 280 nanometers, so resembling let's say that obtaining a more biomimetic construct resembling the characteristic of the native tissue. And then the physical-chemical characterization combined to thermal analysis led to the definition of the overall crystalline content of the polymer, where we register the higher amount of beta phase compared to the other crystalline phases, with value higher than 90% in the final content. So once again, confirming the effective synergistic, the synergistic effect that can have the combination of the electrical pulling and the mechanical stretching during the electrospinning process. We could also perform some quantitative analysis via the measurement of the indirect piezoelectric effect and the definition of the piezoelectric coefficient that could identify values that are reported to be supportive in the cardiomyocyte activity according to different data reported in the literature. So overall, all these data could confirm the effective electroactivity and bioactivity of our substrate. The mechanical characterization also proved the high flexibility of this material, where the mechanical property register for our PVDF patches were aligned with different values that are normally characteristic for some biomaterials already used in cardiovascular surgery and applications. So confirming again that we can be in line with our targets. So despite all the great advantages that are represented by this material and the positive aspect that we have listed before, for sure, one of the main limitations of PVDF is its intrinsic high hydrophobicity. And as we know, hydrophobicity can be a real issue for an effective cell-material interaction, where cells can be, so the hydrophobicity of the surface can hinder cell adhesion and spreading, while inducing a local inflammatory response. So we tried to explore the possibility to improve the PVDF surface, the hydrophilicity of our patches by exploiting a fast and effective surface modification using radiofrequency low pressure plasma. So the choice of this surface treatment was based on the fact that plasma treatment can be very fast and effective in the in the variation of the material surface chemistry and energy, by exploiting ionized gases without affecting the bulk material. However, the effect of plasma treatment and the different process parameters are quite confused in the literature, especially when we look at polymeric substrate. So based on this, we decided to adopt a multivariate approach in order to test and explore different combination of the different process parameters, looking at different power levels, exposure times, pressure values, and in terms of working gases, since we are working with PVDF, in order to make our surface more hydrophilic, we selected oxygen, possibly in combination with argon, in order to stabilize the overall process. So here, as we can see also from the video in the slide, this very accurate and comprehensive assessment enabled the identification of the best processing parameters, where we identify as ineffective the use of low pressure values and the use of argon alone, while the use of oxygen as a working gas and high pressure always led to a very high hydrophilicity of the surface. But unfortunately, here we need to be very careful in the optimization, in the combination of the exposure time and power levels in order to avoid any modification of both the morphological properties, the overall properties of the patch. So we can see here how high power levels can induce a detrimental effect in the morphological feature of the patch, as well as a decrease in the content of the electroactive phase. And also, if we combine it with longer exposure time, we can also induce an extreme loss of the mechanical properties. So in the end, after this very large assessment, we defined the best option of the best combination of parameters represented by high pressure values, low power levels, and longer exposure times, where the effectiveness of our treatment was immediately confirmed by XPS analysis that were detecting the presence of oxygen groups, the formation of oxygen groups on the surface of the patches compared to the non-treated samples. Moreover, the characterization of the samples also further confirmed the effectiveness of the treatment, registering a very a significant drop in the water contact angle of the surface, reaching values lower than 40 degrees. And moreover, the optimized parameters also was effective in preserving all the characteristics of the polymeric patch optimized. So we could preserve very well the fiber diameter morphology, the crystalline content, and also the flexibility, so the mechanical properties of the patch. And we also found the high stability of the process, where all these properties were preserved over 3 months after the initial treatment. And then we could confirm also the higher biocompatibility induced by the plasma treatment, thanks to the collaboration with 2 institution partners, IFRIES from Porto and Newcastle University, that performed a comprehensive biological assessment both in vitro and in vivo that could highlight the improved biocompatibility and the enhanced adhesion in vivo to myocardial tissue. We also could observe from the images how the plasma-treated patch supported a reduced inflammatory response compared with the untreated control. So this was a very large study, so I would like to invite you to read our recent publication on biomaterial advances and to want to get some more details about it. So let's move to address another important aspect of in the design of our multifunctional patch. That is the opportunity to deliver multiple therapeutic agents in order to target the different phases of the regenerative process of the cardiac tissue. So as Karen already mentioned, we selected 2 different drugs, and ibuprofen and pirfenidone was then selected as anti-inflammatory and the antifibrotic drug, respectively. So the incorporation of these drugs into the final patch was explored using different strategies. Here we have just listed the most effective ones that were related to the design of ultrasound responsive carriers loading the anti-inflammatory drug ibuprofen, but also in parallel we explore the effective incorporation of the drugs and the simultaneous release of them in using uniaxial and coaxial polymeric nanofibers, so just directly incorporating the drugs into our polymeric formulation. And then the different successful strategies were then combined in order to enable the multiple drug release from the final patch. So for sure, one of the most well-known nanomaterials that are currently used as drug carriers is represented by mesoporous silica nanoparticles. So these nanoparticles can be easily produced using a Solgen synthesis. So basically just exploiting the hydrolysis and condensation of the silica precursor around surfactant micelle templates, which are later removed to create an ordered porous structure, as we can see here from the schematic. So these materials represent a very strategic nanomaterial for the incorporation and release of different biomolecules and drugs, thanks to their really good characteristic, like the very high specific surface area, the large accessible pore volume that has a diameter ranging between 2 and 8 nanometers, and also the thanks to the high surface concentration of hydroxyl groups exposed on the surface that can allow easier surface modification and drug incorporation. But despite the great advantage and the good properties of this material, if not properly engineered, this nanosystem can lead to an uncontrollable and burst release of the cargo. So in this case, we decided to engineer a stimulus-responsive polymeric coating to promote the retention of the drug and then an on-demand, following on-demand release of it. And in this context, ultrasound represents a very promising external stimulus due to the fact that we can control it as an external control stimulus, external stimulus that can be controllable, and then we can also provide some additional therapeutic effects to biological tissue. So in order to obtain this ultrasound responsive character, we decided to use alginate coatings, to apply alginate coatings to our mesoporous silica nanoparticles in order to promote the trigger release of the drug upon application of an ultrasound stimulus that can weaken, reversibly weaken the ionic bonds between the polymeric chain, acting as a sort of gate that open and close the coating, enabling the release of the drug on demand. So after producing our mesoporous silica nanoparticle by sol-gel technique, we then proceeded with the functionalization of the surface with amino groups, exploiting a reaction with APTS in control condition in order to expose the amino groups on the surface and induce a better deposition and of our alginate coating. And so, as you can see here from the morphological and physicochemical characterization, we were successful in the design of nanoparticles with a size of around 120 nanometers, characterized by a very mesoporous structure with a narrow pore size distribution. And then we then loaded our anti-inflammatory drug, ibuprofen, by exploiting the incipient wetness, and so just incubating also our particles in solution of ibuprofen in ethanol. So at this stage, we then, as a final step of the protocol, we then immerse our ibuprofen-loaded nanoparticles into alginate solution, where we exploited the chemistry of carbonimides in order to activate the surface, and where we tested also different alginate concentration in order to obtain the best performing coating. And then we induced the cross-linking of the polymeric coating using calcium chloride. And here, these are the images that are confirming the successful coating of our particle, registering a slight increase in the final dimension of the nanoparticle, and also the TM images can show the creation of this thin polymeric layer on the mesophoros structure of the silica nanoparticles, and also physicochemical analysis show the drop in the specific surface area and the total polygon volume after the application of the coating. So the last part was the assessment of the ultrasound responsiveness of this carrier. For doing this, we took advantage of the use of a very effective ultrasound platform that was specifically designed by our colleague and partner at Scuola Sant'Anna of Pisa. Specifically, this system as a great represents a very precise and controllable system. So we this enable the precise control of all the different simulation parameters. And so here is the table representing all the different combination of different stimulation parameters that when tested and combined in order to define the best promising and the most effective combination that could actually induce an increase in the release of the drug upon the application of the ultrasound stimulus, also after different repetition of these mechanisms. So here, as you can see here from the slide, at the different time step, we have the boost of the drug just upon the application of the ultrasound stimulus. The developed nanocarriers were then incorporated in our polymeric patch by following the previous optimized protocol. So the carrier were directly incorporated in the polymeric formulation and then processed by the optimized electrospinning process. During the optimization of the procedure, we could define the highest concentration of nanocarriers in the system that could maintain the overall patch properties in terms of morphological feature as well as mechanical and piezoelectric properties. And then the release test performed on the patch sample with and without application of the ultrasound could also confirm the preservation of the ultrasound responsiveness of the carriers also after their incorporation in the polymeric fibers. And here, again, all these data results that are results of a very large study have been recently collected in our publication or RSC advances. So once again, I would invite you to revise and read our publication in case you are interested in more details. So in parallel to this on-demand release of the drug, we also explored other strategies. And so basically the direct incorporation of the drugs into the polymeric fibers, exploiting the direct mixing with the polymeric formulation using both an uniaxial electrospinning setup and the coaxial electrospinning setup with an attempt to better modulate the release profile of the two different therapeutic agents. So starting from the uniaxial system, we basically introduce both the drugs into the polymeric formulation by an adaptation of the initially optimized protocol in order to confirm in order to promote the proper solubilization of both drugs and polymer. And then the formulation, the mixed formulation will then process by uniaxial setup, again adjusting the process parameters in order to obtain a stable deposition of the nanofibers. So here from the images representing the drug release test for both ibuprofen and pirfenidone, you can notice how we decided to test increasing concentration of the drug in order also to define the highest concentration of both ibuprofen and pirfenidone that again could induce the preservation of all the other properties of the patch. And so it's quite evident also here from the release how this optimized methodology also supporting a modulated release of the drug according to the initial drug loading. But then we wanted to explore if we could have the possibility to better modulate the release of the drug. And so in order to obtain a faster release of the drug incorporating the other part of the fiber and a more prolonged release of the drug incorporated in the core, just exploiting the coaxial electrospinning setup. So here we the first assessment was performed introducing pirfenidone in the core and ibuprofen in the other part of the fiber, using the same concentration in order to better understand the influence of the distribution of the drug into the fibers for the further release of both drugs. So here, the optimized process led to the successful design of coaxial fibers that could retain the main properties of the uniaxial PDF patch previously described. And also, as you can see here from the release test performed with the drug, actually the internalization of pirfenidone into the core of the coaxial fibers could induce the reduction of its release compared to ibuprofen. However, no significant enhancement in the release modulation was verified compared with the uniaxial fiber. So we couldn't obtain a modulation of a faster and a longer release of the drug. So there was not a real advantage in the use of this system. For this reason, was not selected for the definition of the final patch prototype, but we are still that is a strategy that is still under investigation for its potential, especially with the use with other polymers and other applications. And then, in addition to the anti-inflammatory drug ibuprofen and the antifibrotic drug pirfenidone, we also aim to target we also wanted to improve the cardiomyocyte proliferation in our regenerative tissue. And this was done thanks to the work of our colleague at University of Coimbra that were developing a mRNA incorporated extracellular vesicles that were further loaded into a hyaluronic acid hydrogel. So this will be the subject of the potential subject of some upcoming webinar on Reborn technology. So I won't give you all the details about this work, but just the main achievements that led us to the successful integration with the final polymeric patch. So here, the strategies involved the different steps where the hydrogel, the hyaluronic acid concentration was optimized, and then so the overall polymeric formulation was adapted in order to better combine with the fibrous polymeric matrix that was then lyophilized in order to obtain a matrix that would be flexible and could be stable over time, so for the long storage of our device. And as you can see from the images, the optimized formulation proved to preserve all the fibrous morphology of the final patch, as well as the overall elasticity. So all these evidence and insight and all these data were then taken into account in order to define the two main prototypes of the patch that you can, in order to have a perfect balance between the effectiveness and the easy translation of our device. So on one side, we have the Reborn patch prototype 1 that enabled the multiple release of the two different drugs, also exploiting the effect of the ultrasound, not only in the biological tissue, but also for the on-demand release of the anti-inflammatory drug in order to boost the effectiveness of our device. On the other side, we also define a second prototype that is represented by the uniaxial fibers containing the mixing of the two drugs, again, in this case, to promote an easy translation of the final device. The definition of the final drug contents is also based on previous results that were obtained in the frame of in vivo drug efficacy tests performed by our colleagues at E3S of Porto, where the specific therapeutic windows were defined. And also, of course, both the prototype can be integrated with the cardiomyocyte proliferative factor, thanks to the use and the combination with this hyaluronic acid hydrogel. So we are now ready for the further in vivo assessment, where we, for both the prototypes, where we have already, thanks to the support of our colleagues at the University of Porto, we could already able to define the properly suturing and the proper adhesion of our patch prototype to the cardiac tissue, as you can see from the video and the images here in the slide. So now it's time to perform the first in vivo test that will define the best performing prototype between the two defined. And then based on the results obtained using the small animal models, we will define the prototype, the unique prototype that will be further assessed on large animal models for the safety and efficacy, finally defining the effectiveness and the performance of our final Reborn patch. So with this, I have concluded, but before actually concluded, would like to thank all the components of our research group, all the partners of the projects, and also all the colleagues here at Politecnico di Torino who supported us in this very large and challenging study. And of course, I want to thank you all for your kind attention. So,
the audience.
okay, I don't know if we can see if there are some questions from the audience.
Or the in-person audience. If you have any questions, maybe they can write them down in the chat.
In case you have other questions that come up in the future, we are always available, of course, for replying to all your curiosity and doubts.
Questions?
No question also here from the audience.
Okay.
So I leave the floor to Andrea.
Yeah, but just a few final things to say. It's just to thank our coordinator, Professor Chiara Vitale Brovarone, who introduced the Reborn project, and to you, Giorgia, thanks a lot also for the organization. It wasn't easy to find the right time to have everyone present, and thanks to the people who are here and also who are connected online. We have a lot of communication channels. We have LinkedIn, which we have been using a lot, and it's growing a lot. We have YouTube, where we also have the Reborn project video that we previously saw, but also other videos with the demonstrations of the device designed by our project partner. I think that's it. We also have the website. You can have access to all the publications made so far, and other publications that will be uploaded later on, but also more information about the consortium, the partners, the news, all the events where the Reborn project took place. It's been 3 years, so it's quite a long time, and a lot of results and dissemination has been done and achieved. I have not much more to say. I don't know if you, Chiara, want to say something more. Okay, Giorgia?
Thank you for the participation.
Yes, thank you all for participating.
And other 2 at least webinar will come, so follow us and you will find info on our website.
Okay. And this is going to be recorded and it's going to be shared on YouTube. That's it. Thank you. Thank you very much to everyone at home. Thank you.
Bye.
Okay.
Vediamo.